Now we are going to pile up what we have to know about cancer with how it is diagnosed and detected and the treatment that we have. Not everyone who gets cancer dies of it, because we have certain treatments for it as well. So there is a ray of hope, and with molecular biology coming into the scenario, there is a ray of hope, greater ray of hope, that we can detect the susceptible genes as well, and then advise the people who are more prone to certain diseases, that don't smoke or don't go and come in contact with radiation. Such things could be taken care of. But before that, we have to talk about their detection. What are the various methods in which the cancer can be detected, and why do we dedicate a lesson to this? Because early detection of cancer is the key. If cancer is detected at earlier stages, it could be cured. So we see, what if early detection of cancer has taken place, and how can we treat it? How is cancer detected? It is conventionally detected by the process of biopsy, where you take out the cells and see if they are growing, then you culture them and observe it. If they are growing rampantly, that means they have lost the contact inhibition property, culture is made, that is known as biopsy. Okay, that is one condition. Apart from that, we have methods and techniques like MRI, magnetic resonance imaging. We have the techniques like X-ray, where we are going to see what is inside the body. We have computerized tomographic scans that could tell us what lies inside the body, if there is any unwanted growth. Apart from this, we are nowadays having molecular biology diagnostics as well. All right. Now, this molecular biology diagnostic includes ELISA, kits like ELISA, where what happens is you check which antigen is present inside the body by providing the antibodies. So antibodies in molecular biology diagnostic techniques are used to see if the antigen is present inside the body. Okay. Apart from this, nowadays we have the very important polymerase PCR technique, PCR detection technique, which could be used to see the condition where the cancerous growth could be occurring. You are going to see the defective gene itself. Okay, identification of the defective gene would be made, and we are going to see what that gene is prone to undergo if it is subjected to certain carcinogens, and people could be advised to stay away from them. So that is also one preventive measure as well as the detection technique. So in this way it could be detected, and if detected at early stages, how it can be treated? We have certain lines of treatments, like simplest and the easiest, if the cancer is not that stubborn, is surgery. Like, right, surgery, that you very meticulously get rid of the tumorous part, remove it if it is tumorous. But apart from that, we have the technique of irradiating the tumorous cells with lethal radiations. Okay, so we have radiotherapy, which covers the technique where the treatment is aiming at getting rid of the tumorous growth by giving lethal radiations. Apart from that, we have another method, that is chemotherapy. Poisonous chemicals would be used to kill the tumorous cells, taking very well care of the cells nearby. So that is chemotherapy. We have radiotherapy, and at times the patient is to be given all the three in order to avoid cancer. So these are the various lines of treatment. Apart from this, there is another treatment that is known as giving the body alpha interferons. Okay, medicinal treatment. Medicines are given in these also, but in the case of alpha interferons, what we use against cancer is our own immune system. While studying about cancer, I don't know whether this came to your mind or not, but there is a question that arises oftenly, that see, the cells will be detected when the cancer has reached so and so stage. Okay, that detection would be possible only if it shows as a lump, although in molecular diagnostic techniques it is different. what happens to the immune system? The body knows everything, that this is wrong, this is antigen. Why does not immune system take care of cancerous cells? Had it been the immune system which is taking care of the cancerous cells, this would not be termed as the biggest killer, deadliest one. Now, the immune system itself is compromised by the cancer cells. You might be asked that why immune system is not able to detect the cancerous growth of cells in the beginning itself and get rid of it. Instead, when it becomes so lethal, or it almost overtakes it, subsides the entire system, it comes into scenario then. So, this immune system, that part of the immune system which detects whether the cell is cancerous or not, is deceived by cancer cells. Cancer cells are very clever in deceiving the immune system. Okay, so even if the immune system is able to detect that there is the cancerous growth, it deceives very well. Okay, it takes over the machinery of the cells which are going to come and kill over there. They are deceived very well, and the immune system could not work. Now, in order to overcome that deceiving of the immune system by cancerous cells, the body is often given alpha interferons so that the deceiving of immune system could be curtailed. Okay, the cancer cells are deceiving the immune system, because of which immune system is not able to take care of the cancerous cells. Now, what if the alpha interferons which are given externally, they are externally advised and inserted into the body, and they make the immune system more active so that they cannot be easily deceived by the cancer cells, is one line of treatment that is given to the cancer patients. Okay, so this all you have to remember. Do keep in mind about this point, that immune system cells are deceived by the cancer cells, but if we administer the patient with alpha interferons externally, then what happens is the immune system becomes very well aware of where cancerous cells are, and it acts accordingly instead of getting deceived. So, this is all about